So today I want to talk about genomic selection and how maybe somewhat more applied talk about how you could actually leverage genomic selection and genomic selection techniques for optimized breeding gain. And I think I'm trying to show you that to do that, you have to work and consider the optimal use of phenotypic data for breeding. I would like to start with something, maybe a basic question, maybe definition. You probably all heard of genomic selection, maybe too much to your own taste, but it's not always really clear what we mean by genomic selection. And I think it's important to have maybe a broader definition than what you might have heard of. First, really simple, genomic selection is the use of genome-wide marker. It is high-density marker covering the whole genome. And you want to use those whole genome marker to increase the accuracy of selection. That is, make more precise selection, maybe try to increase breeding gain, reduce breeding cycle by making selection decision earlier in the breeding cycle. And this applies obviously for unphenotyped individual for which you only have marker to make a selection decision. And most of the paper about GS in plants concentrate on making decision for unphenotyped individual, but it's also true for phenotype individual. It could be for individual for which you have phenotype on some trait, but not other trait. So you could combine phenotype plus marker on other trait. Or it could be also even marker for which you have genotype for which you have phenotype, but low quality phenotype. So you could combine phenotype on the candidates themselves plus marker to increase the precision of selection. And I think it's really important to bear in mind that marker can bring something at all points of the breeding scheme. And I think it linked pretty well to the second issue, why we need to work more on the optimal use of phenotype. This graph presents the cost per raw megabase of DNA sequenced as surveyed by the NIH. And you will notice on the log scale show really dramatic decrease in genotyping cost or raw sequencing cost over the last 10 years. And we can expect those costs to keep going down over time, while the cost of phenotyping should remain the same or maybe even increase because of land use cost or labor cost, which means that phenotyping is quickly becoming the new major roadblock for breeding purpose or for genetic studies. And so we need some kind of a change of perspective, because previously marker was the most expensive part, the most difficult information to generate, so that phenotype was focused on trying to make the most out of the few markers that were available. While in the context of cheap markers, we should instead try to make the most out of the scarce phenotype available by using marker among other techniques. Now, I think it's an important change of perspective that can be made better described in the context of a simple breeding scheme. This figure presents a classic scheme that would probably apply, for example, to wheat or maize, to simple crops, with 4 major steps. We'll start obviously by making crosses and generating inbred lines, maybe through double haploid techniques. Then move on to some kind of early stage testing, focusing on high heritability traits such as height or flowering time, and maybe use some marker-assisted selection techniques for major QTL. Then selecting some entry, moving on to a first set of yield trial that is focusing now on lower heritability trait, some few plots per entry because of the large number of entries. And then moving on to advanced testing with still focusing on low heritability trait, focusing also on traits that are much more expensive to phenotype, such as quality traits, and as well as much more replication, much more location previous to release. In the early day of GS, and I mean also in the context of what we call marker-assisted recurrent selection, we focus mostly on what I call rapid cycling GS. That is, you want to use marker techniques to bypass most of the cycle, that is, go faster and maybe cheaper, so that you use your GS model or your marker to predict the entry really early on, maybe when you might have even just 1 seed per genotype, so that you can make a selection, advance some of the entry, and recross really quickly those selected entry to generate new candidates. So most of what we've focused on so far in genomic selection in plants has been around making those scheme work. But in the context of expensive phenotype, there's a number of other ways we can use marker to increase breeding gains. And some of them are presented on this figure. First, at the candidate generation stage, we could use GS model or marker techniques to predict which crosses are the most promising, that is predicting crosses variants, for example. On early stage testing, we could combine phenotype on high heritability trait with marker data to predict them for yield, for example. So we combine genomic selection hard to phenotype trait plus phenotype on easy and cheap to phenotype trait. We could, when we move to the first field trial, we could combine marker on the candidate plus phenotype on the same candidate to increase precision of selection. That is, combine marker and phenotype. And moving on to advanced testing, we could also use genomic selection model to improve accuracy for every expensive to phenotype trait, such as end-use qualities that are usually not assessed until the very last stage of the breeding program because of cost. And we could also use genomic selection techniques to predict G by E. In this talk, I'm going to focus on a few aspects of those different opportunities offered by GS for breeding programs. Starting with something relatively obvious. To make a selection with GS, you need a model, a genomic selection model. And GS was enabled in the first place by new statistical techniques that I'm going to describe really quickly. First, the basic idea of GS is to try to use all markers simultaneously in a single step. That is, avoid a marker selection step, or variable selection step that's problematic if the trait is highly complex with a number of small effect QTL. Obviously, it's an issue because it means that there is much more effect to be estimated than there is observation, so that classic statistical technique won't work. You need a workaround to develop alternative strategies to do so. And they fall basically in three categories. The first one will be what I call penalized regression, such as base A, base B, the lasso or regression that you might have heard of. The idea being that the underlying hypotheses are different, but you add a penalty parameter to account for the fact that you don't have enough degree of freedom to estimate every marker effect. The second category of model, there are models without marker effect, which is quite related to the first class of model, are models such as GBLUP or LKHS. The idea with that class of model is to use marker to estimate the covariance between individuals, for example, through the kinship based on marker, and use that information about the covariance in a mixed model to make a GS prediction. And it's sometimes overlooked that you can actually do GS without going through a marker effect generating step. It's pretty equivalent to the first penalized regression type. And then we have maybe a broader class of method, machine learning techniques, such as random forest, neural network, support vector machine, that have been tested for GS as well. Got some interest because potentially they could capture non-additive effects that are potentially of interest for breeding. For breeding, what we're interested in with genomic selection model is how accurate we can get, because even a slight gain in accuracy means basically free breeding gains if it's just more fancier statistical methods. And so because so many GS models have been proposed on the early days of genomic selection research, we wanted to test them and we did that on 8 different data sets, wheat, maize, and barley, going from biparental population to diallel to more panel type of unrelated line. And basically the bottom line when we look at cross-validated accuracy for those different traits on different data sets is that you don't see much difference. The most classic one, ridge regression, is more or less at the same level of much fancier methods, with maybe RKHS, we capture some non-additive effects slightly superior. But basically, you don't see a really dramatic picture on average, and it's also true when you go at individual trait level, even for trait with really contrasting genetic architecture when we know there is major QTL. This has maybe two implications. I mean, it means that if you want to do breeding using GS, you don't need to spend too much time playing with GS model because you won't see much difference. So it's easier for practical purpose. It's also kind of disappointing because so much effort went into developing those models. You would hope that they actually deliver something, but apparently not much beyond the publication. And those results were also confirmed by recent simulation and theoretical work that show that with breeding data sets of the size we have, that is far less observation, far less phenotype data than we have marker effect to estimate, we don't expect to see much difference between genomic selection model. And we don't expect to see much difference in prediction accuracy, but also in how those models actually capture the genetic architecture. So beyond the fact that they have similar predictive accuracy, we're interested in whether those models could actually capture something different. Because the underlying hypotheses are so different, maybe they were capturing different genetic signal in the different trait and data sets. So we tested that by using the cross-validated value for every trait on every data set. and use those predicted values to cluster genomic selection methods to trying to find similarities and dissimilarities between genomic selection methods. And the results are presented on this dendrogram. You will note two clear groups of methods. One group here that with random forest, neural network, and RKHS that correspond to methods capturing potentially non-additive effects. On the other side, all the penalized regression type methods. And you will find regression here, Bayes CPI, Bayesian lasso, some kind of Bayes B. Yeah, so not basically, it suggests that if you want to try still some GS model, you might want to try with regression, but not Bayes CPI. Or if you try Bayesian lasso, maybe don't try Bayes B. It's kind of it gives some clue what you should be trying out if you want to reduce the time you spend or the computing time needed. Because it seems that there were a difference between GS model, we tried to combine different GS model, combine different prediction to increase accuracy, but we were not really successful with that. Which doesn't mean it can't be done, but we tried a number of different ways of doing so, and we couldn't get something better than a single GS model, basically. Moving on, if we have a genomic selection model, we still need data to be able to predict. GS potentially unlocks the use of historical data generated by a breeding program, which is something that was not really possible or practical in the past, which is an interesting feature. It also comes with a number of questions because probably not all historical data is useful for breeding. At least we can make that hypothesis. And the main limit to how useful historical data can be is the problem of genotype by environment interaction, or G/E. That is the fact that genotype planted in different environments can react differentially. And it can happen in 3 different types of cases. You could have a case that's kind of ideal with no G/E. That is a linear response of the genotype to the environment. So pretty simple, easy for breeding. You can also have what we call non-crossover G/E. That is, there's a difference of scale between environment, but the ranking of genotype is not affected. So it's not too troublesome for breeding. And then we have a worst case scenario, which is also the most common, what we call crossover G/E. That is change of rank of genotype, which means that if there is no one best performing genotype everywhere, which is problematic for selection. And for GS, the implications are quite straightforward. If you want to breed for environment 2, but some of your historical data contain environment 1, and you don't account for it properly in your analysis, you can actually decrease your breeding gain toward better performance in environment 2. So characterizing well G/E is critical for GS and phenotypic selection. And it might be better explained by the concepts of the TPE. The TPE is a target population of environment, that is the mixture of different environment type that forms the breeding target. It might be, for example, for wheat in New York, we expect that there is different stress type with different frequency of occurrence. And it's somewhat summarized by this figure that presents the performance, for example, yield on the x-axis and the frequency on the y-axis. The orange curve will be the distribution of the traits on the whole target population of environment. But more careful analysis will reveal that there is actually different environment types, 4 of them represented here, with specific frequency of occurrence that are characterized by specific stresses. And we can show that in the presence of G/E, it's critical to adequately sample the TPE to realize breeding gains. So that you bring the right direction and bring for the right stress. So that it's really critical to sample adequately the TPE. And you can see that it's probably more important with GS than with phenotypic selection, because in the context of phenotypic selection, phenotypic data has a limited shelf life use. So if a sample of phenotypic data is a bad sample of the TPE, it's going to impact the breeding gain for a few years. Whereas with genomic selection, because you're going to use the data over and over again, it might have a much larger impact and might be much more detrimental to breeding gains. So for GS, it's critical to make sure that the data we use is actually a good sample. It actually corresponds to what we're trying to breed for. At the same time, we can try to use GS to characterize environment, because we can use the replication of allele rather than the replication of genotype, which can help a lot analyze large unbalanced data set similar to those that are generated by typical breeding program. And we tested that using a large spring barley data set provided by Lima Grain, about 1,000 inbred lines planted all over Western Europe about 14,000 plots, and extremely unbalanced with about only 18 genotypes present in more than 50% of the environment. So pretty typical of what you could get if you have access to commercial breeding program. And really difficult to analyze with classic mixed model methods because there's a number of numerical issues that pop up when you try to fit model globally on the whole data. So as a simple workaround, we considered that the data in each environment was actually a separate data set, and use each data set to generate marker effect, and use that table of marker effect in each environment to try to cluster environment and identify pattern of similarities and dissimilarities between environments. Results are presented on this heat map, which is a clustering based on marker effect using Euclidean distance, with bluish color indicating more similar environment and reddish color more dissimilar environment. You will note we have 4 outlier environments, or environments that appear to be outlier over there, that were really clearly picked, and that signal was not obvious at all based on the phenotypic data. So we were kind of excited about it, and it seems that at least for one of the environments, the trial was planned too deep, so we probably picked that signal, and maybe a heuristic explanation of why we're picking outliers so well is because the metric we use give more weight to large marker effect. And if you have an unusual stress, a stress that creates large, that you have never been breeding for, that is an outlier stress, you might expect large QTL to show up in your population because you never selected for it, and or metric somewhat picks that up. So what we could also do, because we have a genomic selection model for each environment, we can predict accuracy from one environment to the next, get basically every pairwise accuracy using one environment to predict all of the other. That give us a proxy for the genetic correlation between pairs and environment, which is a critical information for breeding, because that level of correlation actually can be used to assess whether it's useful to sample of the environment, or if you should have different breeding targets. And the results are presented still on this heat map, still with a reddish color indicating more similar environment, that is higher genetic correlation, and bluish color more dissimilar environment. The black box on the top indicates the outlier environment we identify with a marker, so they don't show up here anymore. And the picture is obviously not as clear as with the markers. There's much more noise, probably because in each environment we had very few replication, in the sense that there were a lot of error in the estimation of genotype performance. But still we might find two groups, not very clear, two groups of environment, which is somewhat disappointing, but at the same time we know from commercial barley breeding that usually best performing line in Europe do well everywhere, from the UK to northern France to Germany to the Netherlands. So somewhat expected. Just to maybe summarize this part, I hope I convinced you that the understanding and knowing what the TP you're breeding for is critical for GS, even more so than with phenotypic selection. But that genomic selection, because you can use and leverage the replication of allele, give you some leeway to investigate and characterize the GBI pattern in historical data. To go further with that, we wonder whether we could predict genotype by environment interaction using genomic selection model. And if we can predict GBI, we can potentially investigate better the TP, try to get down to different environment type and the different stresses. We can also potentially predict genotype stability, which is critical for commercial success of variety. And we can maybe also potentially identify critical stresses, and that information can be used for better breeding and more focused breeding effort, for example. It sounds like a good idea, but we quickly run in what we call in the lab the Anna-Karenina effect, from the first sentence of Leo Tolstoy book. That is to say, maybe in simpler terms, that any number of stress occurring during the crop cycle can generate GBI. Whereas if you don't have GBI, it means that there were probably no stress, which is a much simpler case. It's much easier to predict a no GBI case than a GBI case, because anything can happen from planting to harvest, especially on a crop such with a really long crop cycle. At the same time, we know for winter wheat which stressors are important when, from a wealth of agronomy knowledge that we usually don't tap as breeders. We expect, for example, that low amount of light during meiosis will cause kernel abortion and decrease yield, or that hot temperature during grain filling will still be detrimental for yield prediction. So we somewhat, even though we expect any stress occurring during the crop cycle to be detrimental and to create G by E. We still have kind of an idea of what's important when, and we can try to use that in a genomic selection context to predict G by E. To do so, maybe the easiest way of thinking about it is try to consider QTL as a function of environment variable instead of seeing QTL as a constant. And there is kind of 3 limits to it, trying to model in QTL as response curve. The first one is the high dimension of the data. If we just look at, for example, the minimum daily temperature along the crop cycle for one site, we have a really high number of data points, probably each data point contributing really little to genomic selection to prediction, contributing really little to G by E. So how do we leverage and try to simplify that problem? The second issue, which is linked to the first one, is the high correlation of variable. That is, if you look at the correlation, autocorrelation of minimum daily temperature along the crop cycle, you will know that if you take one given day during the crop cycle and minimum temperature 1 month later, the correlation is still around 0.4. So it's kind of, I mean, you have a context with a lot of variables, each contributing really little to prediction and highly correlated with each other, so really difficult to deal with. But at the same time, genomic selection offered us a number of techniques to work around that problem of high dimensionality and high number of correlated variables. The last issue, which is specific to the problem of G by E, is that we don't expect realistically the plant to respond linearly to the environment. For example, if you look on a classic physiology textbook, what a classic plant growth over time is, it's definitely not linear. And we might at least make the hypothesis that QTL should not have a linear response curve to any environment stress. We expect threshold effects or any kind of really complex response surface. So we need to be able to capture some of that, nonlinear responses. At the same time, agronomists have developed a number of tools to deal with that problem of high dimensionality and integration of daily weather input and the problem of nonlinearity of response. And those models are called crop model. A crop model is just a set of complex equations modeling how the plant is going to respond to the environment, how the plant or the canopy is going to develop, and are really simply presented on this figure. Those models will take as input radiation, temperature, rainfall, management practices, and predict how the plant is going to develop a leaf canopy, include feedback loop for depletion of water from the soil, also include prediction of development of phenology, develop leaves, predicts development of leaves, predict development of flowers, predicts development biomass, and finally predict yield. So maybe a simple and not as simple, but elegant solution would be to try to use crop model to make prediction and work on crop model parameter, that is the parameters that distinguish between one genotype to the next, and use those as straight, hoping that they will have higher repeatability, less G by E, and will be basically easier to work with classic quantitative genetics tool. There is 2 limits to this approach. The first one is that it's not clear at all whether crop models are sensitive enough to distinguish between 2 highly selected genotypes. I mean, there is really conflicting report in the literature about it. So we might not be able to pick the small difference that make one genotype be best performing in the field compared to a basic one. And at the same time, if you want to work on crop model parameter, you need really specific phenotyping that are extremely expensive to collect and that nobody cares about, basically. So you can't use existing breeding data to study G by E. So it might work if you have a couple $1 million budget, but on a public sector context, it's a bit tricky. So what we tried to do was to try to consider the crop model as pre-analysis tool as a way to crunch the data on the first analysis step to leverage the fact that they're really good at integrating complex input at predicting development stages. And we do what we call a physiological integration of the environment data. The idea is that we use the crop model to predict the different development stages because crop models are really good at that. And because we know which stresses are important when, we can generate stress covariate per stages. For example, we'll have a stress covariate that counts for how many days during grain filling the temperature was above a critical threshold to count the duration of each stress, for example. We could have a number of stress covariates that account for drought stress per stages. So that and this has a number of advantages. First, we use crop model for what they're good at, predicting phenology. We leverage agronomy knowledge because we can use, by doing so, what stressors are important when, and there is a lot of literature around those issues. We reduce dimensionality from daily weather data to a couple dozen covariates, which make our job much easier. We can use existing breeding data because we don't need fancy phenotype to be collected. We just need environment data for each trial and probably a planting date to fit to the crop model. And also because the model is grounded in agronomy knowledge, we can actually interpret and make inference about which stresses are important and maybe talk a bit more about the biology of GBA and stress response. At this point, we're only halfway through the modeling, in the sense that we have stress covariate, we reduce the complexity, but we're still not modeling QTL as a function and environment variable. And I'm going to focus on that in the next few slides. I know not everybody is comfortable with equation, but I think it's still important to discuss how we could actually do that and try to be as clear as possible. The simplest way of doing so, integrating external input in GS model or in any kind of plant breeding data analysis, is what we call fractal regression. The simple model is presented here. If you don't look at that term, the rest of the equation is a simple trial analysis by mixed model, with Y being a response variable such as yield. We have a grand mean. We have beta will be a fixed environment effect. U will be a main genotype effect. And the originality here is that we have gamma that are genotype-specific sensitivities. That is a linear response of every genotype to every stress covariate, with S as being the matrix with the score of every stress covariate in every environment. We can extend that model further on a GS context by considering, instead of genotype-specific sensitivities, marker-specific sensitivities. And those two models are exactly equivalent, at least in their formulation here. So that now we have a main marker effect alpha, V being the marker matrix score. That is the score of every marker for every genotype. And we have phi that are marker-specific sensitivity to every stress covariate. So it's a pretty appealing model, but in practice, it's almost never been used with more than one stress covariate, and usually only for QTL mapping, because the problem is extremely highly dimensional, because you have as many predictors to calculate as you have marker. So if you have high marker density and the number of covariates, it blows up pretty quickly. So as a kind of simple workaround on that, we use the result I presented for the Barley data. That is, we could calculate marker effect in each environment. And if we have marker effect in each environment, we can calculate the variance of those marker effects across environment. The idea being that if a marker contributes to GBA, it has to be variable across environment. That is, it's a necessary condition, not sufficient, but at least necessary condition, so that we could use the most variable marker and fit a linear sensitivity to stress covariate only for those. This histogram presents those marker effect variants across environment, and you will know that a few are extremely variable with one marker here that corresponds to PPD1, the most important photoperiod loci in wheat. There was extremely variable effect across environment. So we can kind of pre-select which marker we're going to focus on for prediction. So that's the model simplified down. Instead of having V, a sensitivity for every marker, we have a sensitivities only for a subset S of marker, which make the fitting easier. You also note that now we have a setup parameter in the equation. It's because for numerical reason, it was easier to fit the model in two steps, fitting first the main effect model, main marker effect, and then working on the residual to try to capture some GBA signal. There's a limit to that model in the sense that we don't capture complex response, we only capture linear responses, and we expect from physiology knowledge that the response curve are going to be really complicated and might be better modeled actually by some kind of function f that will take into as input the stress covariate as well as the marker score. And the issue being that we don't really know what is the response curve. We don't have much expectation on what the response curve should look like. We at least expect that it's going to be really complex and really hard to model. So as a worker on that, we use the machine learning techniques, soft-rule fit, that was developed by Denis Agdemir, postdoc in the lab. The idea being that we want to capture complex interaction pattern between marker and stress covariate. And doing so, we want to be able to capture nonlinearity. With two constraints, we want the model to be sparse, that is not too many terms. And we also want the model to be interpretable. We want to understand what's actually happening in the machine learning process. try to keep the black box as small as possible, basically. So I'm going to the next slide in a bit more detail about what is that machine learning techniques. The idea is to generate rules from the data. And the rule is just a fancy word for a binary predictor, that is a predictor that takes value 0 or 1. And we generate those predictors using regression trees. A regression tree is a relatively simple statistical technique that take as input a response variable y and a couple of predictor variables such as x and z. An algorithm for regression tree will identify splitting rules that provide a partition of the data depending on the response variable y, but also on the input variable. So that if you take each leaf of the regression tree, it actually corresponds to a rule, to a partition of the data that say, to belong to this leaf, you have to have x below 0 and z below 1. So that rule will be value 1 if you're there, and value 0 if you're over there. So that each of those rules is actually a composite predictor that you can use further in a large penalized regression steps. So we generated a number of those composite predictors using bootstrap sample of the observation of the data, generated a regression tree parsing the rules. And it has been shown in a number of machine learning papers that that technique is extremely powerful to capture really complex response pattern without specifying a priori what type of pattern we're interested in. So it seems to be a perfect tool for what we're looking for. Next, before going to the result, we had access to really large winter wheat datasets provided by Limagrain, planted from 2006 to 2011 in France, with about 44 environments, going from north to south France and about 2,500 genotypes. And we had a quite low SNP curve rate, about 1,300 SNPs, but at the time it was what was feasible on such a large population in wheat. But we had perfect coverage of the major adaptation loci, such as dwarfing genes, photoperiod sensitivity genes, and fertilization genes. That's the originality maybe of these data sets. We have access to daily weather data provided by the European Commission on an interpolated database. That is, they took weather stations all over Europe and interpolated on the grid what would be the daily weather data at each grid point. So that we could use, given the GPS coordinate of every trial, we could pull the weather data for free, basically, and feed that data into a crop model called Serious Quality to predict the different development stages. And once we had the development stages, we could predict stress covariate, such as number of days with each stress at any point during the crop cycle, or more complex stuff such as drought stress and deficit, water deficit, or lack of light during meiosis. For the result, what we were mostly interested in was to build models that can predict G by E for new unobserved environment for which we only have environment data. And the answer in short is that we beat the Karenina effect, basically. To prove that, we split the data in half, taking 22 environment as training and 22 environment as validation. And we looked at the accuracy, prediction accuracy in each environment of the validation set. Try to see how well we could discriminate between genotype in a given environment. On average, we gain about 11% in accuracy, and we decrease the variance of that prediction accuracy by about 11%. So we're more predictive, and the prediction is more stable. And if you look in more detail, this graph presents the accuracies, the pairs of accuracy for every validation environment, with on the x-axis the accuracy with the basic GS model, what we do currently. On the y-axis, the accuracies when we include the G by E term. Each blue dot corresponds to an environment, and the orange line corresponds to the identity. So basically, if a blue dot is above the line, the G by E model is performing better. And if it's below or on the line, we're not as good as the basic GS model. So you'll know that we're not performing better everywhere, which kind of expected because G by E is so complex. We can't expect to capture all the patterns. But we gained quite a bit, especially for environment in which we were really bad with the basic GS model. So there is some hope, and we could probably do better because by splitting the data in half, the training set is relatively small, so larger data set should increase quite a bit the accuracy. I would also like to point out that it's the first time ever that somebody is reporting prediction accuracy for new unobserved environment. So there is basically no expectation in the literature on how much G by E could be predicted. And probably a lot of people will argue that it can't be done. So it's still pretty exciting. Because we can predict G by E for any environment given weather data, we can predict G/E for any number of genotype and use that to calculate the genetic correlation between pairs of environment. And we can do that with for environment with phenotype, but also for what we call virtual environment that we have simulated using climate change model or using historical data to try to get a better sense of the TPE. And it's what we did here only for the environment for which we had phenotype data. You will note that it clusters mainly by year, with 2008 year, for example, 07, 09, and like here, a small group of environment in southern France, which was quite unexpected because, at least traditionally, we expect to have two breeding targets, a North France and South France target, but we don't really find that here. And it's not really clear why, but it might be, breeders tend to say that the climate tends to be more variable in the last few years, but I mean, quantifying that is a bit difficult. But at least we don't find the pattern, the classic pattern as strong as what we're hoping. And maybe we started to do, but it's not presented here, we can predict G/E for historical data, and we had 30 years of historical weather data, and we had a climate change simulator model, so we could simulate 50 or 100 years of climate with different climate scenario. So we could use that to identify the different stresses on the different environment type forming the TPE, looking back but also looking forward. And that's absolutely a critical piece of information if you want to be better at phenotyping, picking better location for your trial, and try to find the best strategy to sample the TPE. What we could also do was identify important stresses for G/E, and we did so with the permutation strategies, try to find stresses covariates that were significantly associated with G/E. And surprisingly, most of them correspond to stress early in the cycle, not terminal stress, with, for example, drought in early spring and heat stress before flowering and in early grain filling. With both, for each stress, we had two covariates, one covariate for the intensity, that is how many number of days, for example, you have a drought stress, and also intensity, that is what's the magnitude of that drought stress when it occurs, accumulated over the period. So it's quite interesting. And it's also warning in the sense that we don't find terminal stress when we expect terminal stress to be somewhat important, at least once in a while. And looking further at the data, it seems that there were no good summer in Europe from 2006 to 2011, so no terminal stress. So even though our data set was large, it wasn't large enough to sample all the different stress that you might want for G/E. So maybe a really strong take-home message is that bigger data is much better, especially for G/E, and larger sample absolutely critical, going multi-location and multi-years. As a quick summary on this part, I think I've shown you that we can capture some G/E even for unobserved environments, which provide a really powerful tool to study the TPE, but it can also be used to predict genotype stability. And we've shown on the wheat data that the genotypes that tend to be predicted to be high-performing and most stable are also the ones that have been best commercially successful in the last 10 years. So it's hard to quantify how good we are at predicting stability, but at least heuristically, it seems we're doing a pretty good job at that. Overall, I think I've tried to show you that current GS models tend to be equivalent, even though in the lab we're working on new type of genomic selection model that might bring more and try to incorporate different type of data or information to be better at prediction. I think I've shown you and tried to convince you that defining the TPE is critical for GS, even more so than with phenotypic selection. But at the same time, you can use simple genomic selection approaches to quantify G/E and study complex G/E pattern. And also that we can combine the strengths of genomic selection and crop modeling to predict G/E and explore phenotyping strategies and breeding strategies. I think overall it should help optimize the use of phenotypic data for breeding, which makes it really exciting them to be a plant breeder, because as marker data get cheaper, the most valuable part of what's happening in the breeding program is the phenotype, which means that the breeders are again back at the center of what's happening, which really make it a great time to be doing plant research and plant breeding, more than being in the lab generating markers, basically. With that, I would like to acknowledge the funding and the support for Lima Grants, provide data and support and tools, and add the foresight or the oversight, I don't know, to fund my PhD project when I barely knew them. And I would like to thank my committee, Mark, Jean-Luc, Jason Media. It was really a pleasure to work with them. and a number of people at Limagrain and in Mark and Jean-Luc's lab. Thank you.
Okay, well, we have time for a few questions. By the way, the donuts were a special request by Nicolas.
Yeah, I was counting on the sugar high.
And I think that his work on modeling genotype by biointeraction using these environmental covariates is truly a breakthrough. And I think we're going to hear a lot more about this approach in the future. So, any questions out there, Mike?
Great presentation. So, quick question on, in terms of now that you're going to be going into like a breeding company and in thinking about G by E and the TPE, so how would you, I guess, decide on new locations to include in your breeding program? So say you're now in charge of this breeding program in a company, how are you going to, in a clever approach, expand your testing?
So if you assume that you can predict the genetic correlation between any number of locations, just provided with the data, you can calculate those correlations on a grid and try to find locations that tend to sample well the TPE. Or you could also have adaptive strategy, that is, you use your classic scheme. 1 year, you get a sample of environment. You can calculate what you actually sample of the TPE and what's missing. And you can, based on that, try to have basically an adaptive strategy, pick the location for the next year. So that will maximize the probability that you sample, that at the end of the phenotypic testing, you have an adequate sample of the TPE to make a selection decision. But I mean, when you start doing that, you get a number of questions on how you optimized and what is actually the best way to optimize. If you look at those papers from the Australian, Scott Chapman or Graham Hammer, they use clustering, but if you know the genetic correlation, using clustering is not adequate, I think. It's better to try to derive mixed model criteria directly from the mixed model.